say that the animal is facing which way, left or right? Not that I'll ask you that.
Upside down.
No, it's right side up.
No, it is.
So this is the anterior because it has all the red stain, right? And remember what kind of cells make that, what kind of cells stain bright red like that? So if I were to point at one, if I were to go at higher power, let's say, and if I do it, I will always give you some context, which means I'm not just going to show you anterior, I will also show you like maybe a region like this, although these blood vessels kind of get in the way a little bit. But, you know, there's a little more of a field in the microscopes than there is on the video screen. But what I would do is I would show you some context here. And also, you're not going to see any kind of red like this. So if you see the pituitary, if it's a high power slide and I ask you for a particular cell type, it's the acidophils that are bright red and they make growth hormone or prolactin, actually. Most of them are dedicated to one or the other. If it has sort of a bluish cytoplasm stain, those are basophils, which make gonadotropins or TSH. Right? Chromophobes make ACTH or nothing. Right? Sometimes they're chromophobic just because they don't have any secretory granules, rather than POMC. Right? And if I ask you about the posterior, so hopefully, if I show you it under high power, I will definitely provide the context so that you can tell it's the pituitary, which you'll know just from the color. But if I'm pointing over here instead of over here, you'll know that it's not a trophic hormone, obviously. Okay, so that's basically that slide. We didn't really do any specific histology of the hypothalamus area, so I'm not going to ask you about that. All right, so that's basically the question A will ask you to identify the cell type, the gland, the part of the gland, and the part of the gland. Questions B and C will typically be asking you for hormones that the indicated cells make, or hormones that the indicated cells are targets for. So they'll be, functional questions more like that. No hard and fast rule, but basically I'll be asking for like 3 things about each thing it's pointed to. Next is the thyroid. It's not alphabetical. Yikes. There it is. Something. Okay. It's way out of focus on it. Does she, I got to deal with Victoria, about sharing this thing. So she's going to come in and claim it, and then she's going to give it back after she's done with it.
She's done. She should be done by now. I was going to give it to you after 11:10.
No, but she's doesn't have it now. She wants it probably.
No. Her class is done at 11:10, so she should be done. I think her students are leaving now, so she doesn't need it.
Oh, really? Oh, okay. All right. Okay, thanks. Hey, we successfully stole this. All right, so here we have thyroid and parathyroid. Again, I will always give you context. So you can see there's thyroid and parathyroid, remember? So this looks more like a pancreatic islet. I'm not going to show you that. I don't know what that is. But I'll obviously ask you for things that are more typical. The other thing that's often, this is a very atypical slide. And they're both almost the same. But anyway, one thing that sometimes I do have a tendency to get a little tricky with this because you'll see that there are thyroid follicles roughly the same size, and a lot of times you'll see a small artery or a vein. And sometimes I'll ask you to identify the gland. You say, oh, I know that. That's thyroid gland. And I say, identify the structure indicated. You say, oh, I know that. That's a thyroid follicle. It may be a small artery or a vein. So I'll kind of throw that in every now and again because for some reason our thyroid slides often have very good examples of small arteries and veins. And you should be able to recognize what blood looks like. I will never ask you to identify a blood vessel unless it has blood in it. So if you know what blood looks like, which are pale pink cells, that's fine. You can see here this is just basically a solid mass of thyroglobulin, or colloid substance, whichever you want to call it.
So you're saying that blood vessels and colloid fluid have a different color? Like,
well, they're different structures. See, the colloid is basically a solid pink. It doesn't have all those little pink discs. You should know what blood looks like. It doesn't look like that.
the plate, what you're saying is that the red blood cells are observable in blood.
Or what?
Are
Yeah,
observable.
yeah. Blood is mostly red blood cells. So if you see a blood vessel, that's what it'll have in it. But this is what thyroid follicles look like. Typical questions also might be, thyroid hormone is unique for a particular chemical element that you find nowhere else, namely iodine. Speaking of chemical elements, this gland is particularly designed to control what chemical element in the blood? Calcium. Okay. You know, these are all functional questions that could be asked about this tissue. So it's thyroid and parathyroid.
What are the islets?
What are islets?
Oh, sorry, that's...
That's the wrong gland. But another thing, what's the tropic hormone that stimulates these things? TSH, right? So that's another thing that could be done. Also, the purpose of thyroid hormone is controlling the metabolic rate. So all of those things could be asked. What was the red thing? Well, you mean all these things?
Yeah.
Well, that's thyroglobulin or colloid substance, depending on whether you want to call it what the microscopists call it or what the biochemists and endocrinologists call it. This is a little bit. Usually, there's a little more of a gap here, and not so tight. But you see, there's this connective tissue line between the two. And this is from, as you know, when you made the slides, one of the things you do is you put these things progressively through alcohol. You dehydrate. And when you dehydrate an aqueous gel, it tends to shrink. So that's why you have these white spaces here are artifactual. So they're just, the colloid just, thyroglobulin, as it were, just shrinks. So anyway.
So the only cell type here really is the...
No, I forgot. Yes, that's true. There is also the C cells. Thanks for reminding me. I didn't, because none of them, I didn't see any kind of jumping out at me. I mean, here's one. If you see a very bright chromophobic cell that's clearly not in a follicle, that's a C cell that makes calcitonin. So that, you see how that's, and that's sort of clear. It's obvious. It's a nucleus that appears backlit because the cytoplasm has nothing in it that stains, and it's clearly not associated with the follicle.
You talk about the slightly chromophobic one?
It's not, what do you mean slightly chromophobic?
It looks different than the other ones.
Yeah, it looks, you can see that the nucleus looks backlit,
Okay.
illuminated, because this is not the clearest example of it, but there are several here. Usually what I do is the questions are drawn in advance, like what kind of cell type, what kind of hormone, and all of that, and then when I actually get the slide, if something strikes me, like if I happen to get the slide in there and I see this very obvious, parafollicular cell, also known as a C cell or a clear cell, kind of jumps out at me, then and it's very obvious, then I might ask about that. Okay. So, but that was the clearest one that I saw there. And there are a couple of others here. They tend to be kind of large, although that's not totally chromophobic, but it's slightly chromophobic.
They don't necessarily appear in clusters.
No, they don't necessarily appear in clusters. Sometimes they're isolated, but like here's a cluster of a couple of
They're C cells, you said?
C cells, which is short for clear cells, also known as parafollicular cells. They secrete calcitonin, which has the opposite effect of the parathyroid, which is across the river here, in the other gland. But anyway, enough of the thyroid, I think.
Do we have to identify anything in the parathyroid gland? Cell types?
It's just chief cells is really all. Remember I said you can't really find any oxyphil cells.